KURINJI Senior Secondary School, CBSEE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSEE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.com.
Now students, so in this video we are continuing nucleic acids information. So complete detailed information or detailed information now I am going to give you on nucleic acids. Okay, now the heterocyclic compounds in nucleic acids are nitrogenous bases. So here we have discussed, suppose if you take one particular nucleotide, nucleotide consists of three different types of components. One is nitrogen bases, second one is pentose sugar, and third one is phosphate. In yesterday's in previous video we seen no. So now in this video, first of all now we are going to discuss regarding heterocyclic compounds known as nitrogenous bases we are discussing. If you see there, heterocyclic compounds in nucleic acids are the nitrogen bases. So these nitrogen bases we are giving or we are calling by one more name, heterocyclic compounds. Now what are the heterocyclic compounds are there means adenine, guanine, uracil, cytosine, thymine. So total count panninga one by one, 1, 2, 3, 4, 5. No? So adenine, guanine, uracil, cytosine, thymine, isn't it? If you take our knowledge point of view, nucleic acids, two types of nucleic acids, no? What are those? One is DNA and one more is RNA. So nalla save panikunga. So nucleic acids two types. One is DNA. DNA means we know very well now deoxyribose nucleic acid or deoxyribonucleic acid, and RNA means ribonucleic acid. Out of here there are 5 different types of nitrogen bases are there, no? Ange what are the nitrogen bases are present in the DNA and what are the nitrogen bases present in the RNA means once you see now here you see adenine and guanine present in both, uracil will be present only in RNA and thymine only is present in DNA. So thereby confirm panikonga, uracil is present in RNA and thymine is present in DNA. So now here one more important point I will give you. Once you see adenine simply A we can take. Adenine always binds with thymine and guanine always binds with cytosine. Okay, so don't forget. So adenine always binds with thymine and guanine always binds with cytosine, and between the adenine and thymine how many number of hydrogen base or hydrogen bonds are there? No, 2. And between the guanine and the cytosine, no, 3 hydrogen bonds are present. So don't forget, that is in case of DNA. Suppose in case of RNA, how sir this particular bonding will be there means adenine with 2 hydrogen bonds, no, binds with or linked with uracil, AU, and common one is guanine with cytosine common. So means guanine with cytosine with 3 hydrogen bonds is common both in DNA as well as in RNA, but in case of DNA, adenine always binds with thymine, and in case of RNA, adenine always binds with uracil. That's why AT in case of A with 2 hydrogen bonds, or adenine with 2 hydrogen bonds with thymine, and guanine with 2 hydrogen bonds with uracil. Okay, ma? So that kind of information, so primarily we have to discuss. Okay, now next one, suppose here you see there, here totally 5 different types of nitrogen bases are there, no? Adenine, guanine, cytosine, thymine, uracil. Likewise there are different types of nitrogen bases are there. Again in previous video I hope I have given some more information, purines and pyrimidines. Did you hear? Or otherwise no problem, now you will learn. Now nitrogen bases, our convenience to study, now we are going to divide into 2 types. Okay, so nitrogen bases 2 types are there. One is purines and one more is pyrimidines. Here down is there, you see pyrimidines. Okay, now purines, what are the purines out of 5 types of nitrogen bases means? Here we have written in the third line, once you see adenine and guanine are substituted as or considered as purines. Which one? Adenine and guanine. Adenine and guanine combinedly or both together we are considering under purines, whereas rest of the things or the rest are considered or substituted as pyrimidines. Means purines, how many numbers? Number point of view, how many are there? Rendu, adenine and guanine. Bonding point of view, bonding or bond formation point of view, adenine binds with thymine, guanine binds with cytosine. So thereby, you don't know that one. Okay, in +2 definitely I will give you that information. So here adenine always binds with thymine and guanine always binds with cytosine. Okay, so thereby here when adenine and guanine, once you see when adenine and guanine, so when we will be considering as a purines, remaining three we can consider them as pyrimidines. So thereby according to one scientist name here Chargaff. Okay, so according to Chargaff rule, the ratio of purines and pyrimidines will be 1:1 ratio. Just listen it, and suppose if capability is take into your mind, otherwise leave it, no problem. But in +2 you will learn very clearly. So now what is the point now I am going to give you is adenine or purines always binds with the pyrimidines. So thereby ratio of purines and pyrimidines always will be 1:1 ratio in case of DNA. So thereby nitrogen bases 2 types are there. One is purines and second one is pyrimidines. Next one, skeletal heterocyclic ring is called purines and pyrimidines respectively. So there heterocyclic compounds, what are the heterocyclic compounds are there means? So nitrogen bases, our knowledge point of view, we will be considering as heterocyclic compounds. There monocyclic compounds or dicyclic compounds, likewise we have learned information, isn't it? So definitely here the purines and pyrimidines simply we will be considering under heterocyclic compounds. That point here also we have learned. Once you see in first line, so we have started with the heterocyclic compounds only, no? Adida. So now heterocyclic compounds are simply nitrogen bases. Our knowledge point of view, we have been divided into 2 types. One is purines and one more is pyrimidines. Next one, the sugar found in polynucleotides. So from here onwards, important. The sugars are found in, one more time you see, sugar found in polynucleotides is either ribose or deoxyribose. Means here pentose sugar, three number of components are definitely involved in the formation of a polynucleotide. We see no, or simply if you take one nucleotide, there 3 components are present. What are the 3 components? One is phosphate, nitrogen base, and one more is pentose sugar. No, this is pentose sugar only. Now simply we are considering as sugar. Pentose you can remove, so sugar you can call. Okay, so now this particular sugar, if you take there, 2 types of sugars are there. One is ribose sugar and one more is deoxyribose sugar. So then, sir, where we can find out the presence of sugar or ribose sugar, and where we can find out the presence of deoxyribose sugar means, once you see here, the nucleic acid, listen carefully, it is important, a nucleic acid containing deoxyribose idima idida. So a nucleic acid containing deoxyribose is called deoxyribonucleic acid, simply DNA. And while that which contains ribose, next one contains ribose, we will be calling as ribonucleic acid RNA. So now did you find out any difference? Inge one important need bit actually I have to give in +2 standard, but one point I will give you, try to save in your mind. What is that? No, if we are learning nucleic acids, two types of nucleic acids. Now we will think that in comparison of DNA and RNA, no, so first one or first formed one we will think that as DNA, first genetic material. But what is the first formed genetic material? No, that is RNA. Okay, now here RNA is ribonucleic acid, is a first genetic material, and by the evolution, while evolution process is going on, DNA is formed from the RNA. Okay, so thereby here what is the point here we are going to discuss is nucleic acid. So nucleic acids, two types of nucleic acids are there. One is a DNA and one more is RNA. Now DNA is formed from, DNA was formed from the RNA. So nalla save pannikonga important. Ipo diagrammatical information once you see. First one is ribose sugar. Once you see ribose sugar, once you see pentose sugar, pentose means 5 carbon compound. Pentose sugar means the sugar which consists of 5 carbons. That's why we are considering as a pentose sugar. Okay, now here once you see here, it is first carbon, here exactly spotlight, and second carbon, third carbon, fourth carbon, and fifth carbon is outside of the pentose sugar here. Okay, totally 5 carbons are there, no, that's why we are considering as a pentose sugar. Ange second in second position or in second carbon, no, hydroxyl group is there, no, or simply OH group is there, no. So that, but in case of deoxyribose sugar, no, here O not there, you see. Difference can you find out? Once you see what is the difference we are observing between the ribose sugar and deoxyribose sugar. In ribose sugar, no, at second carbon of pentose sugar we are finding OH group, but whereas in case of deoxyribose sugar, no, we are finding hydrogen group only, but here oxygen not there. Sir, what was happened? Why we have written here like this with the OH group, and why here we didn't write the O or oxygen, sir, means? So now only one point I said, no, from the DNA, or sorry, from the RNA, DNA formation taking place, no. So this is the main reason. And DNA we are calling as deoxyribonucleic acid, no, deoxy. Once you see, D means removal. D means removal, isn't it? And which one is removed? Oxygen. Okay, from the ribose sugar, once you see from the ribose sugar, oxygen is removed. That's why that particular ribose sugar we are calling as deoxyribose sugar. Point purida. So now what is the point? No, actually ribonucleic acid only is there initially, but when particular pentose sugar lost the hydrogen at the second carbon of pentose sugar, automatically it leads to the development of, or it leads to the synthesis of DNA. That's why we have given a name as deoxyribonucleic acid. Ribonucleic acid initially, but when second carbon of a pentose sugar loses the oxygen, no, so one new compound formation takes place. That new compound name only we are calling as DNA. DNA is nothing but deoxyribonucleic acid. I hope you understood this point, right? Now let us see the nature of a bond linking monomers in polymer. So means what are the types of bonds are there, and what are the types of bonds are present between the monomers of polymers. Okay, once you see what is the side heading meaning, if you take polymers, polymer means there so many number of components are there. Poly means that only, no, meaning many, means many components are linked together. So now what are the main components, or more number of components are linked together? No, so all together forms the one polymer. So there are many monomers, there are many monomers comes together or linking each other and results in the formation of polymer. Okay, that's why here we kept a side heading like this. Now what are the types of bonds are present in the monomer, or what are the types of bonds are present in the polymers? Now let us see one by one. Now in a polynucleotide, or polynucleotide means simply we can write it as protein, no. So in a polynucleotide or protein, amino acids are linked by a peptide bond. ఇది maximum members కి తెలియం ఏమా. సో మీన్స్ సపోజ్ ఇఫ్ యూ టేక్ వన్ పాలిపెప్టైడ్ చైన్ ఆర్ ఇఫ్ యూ టేక్ వన్ ప్రోటీన్, నో దేర్ అమైనో యాసిడ్స్ ఆర్ లింక్డ్ బై peptide bond which are formed when carboxyl group means when COOH group of one amino acid reacts with the amine group of next amino acid. So this point nalla save pannikungama. Even in +2 also, while explanation of translation process, we will learn this one. Okay, so thereby if you take one protein, or if you take one polypeptide chain, there definitely all amino acids are linked together with the peptide bond and results in the formation of a protein. So now here one more point, that amino acids are linked together by a peptide bond. Amino acids are linked together by a peptide bond and results in the formation of polypeptides. If one peptide irukana, so that one we could not be, or we cannot call it as a poly one. Poly means many. That's why when many mono or when a more number of amino acids are linked together with the peptide bonds, then only it leads to the formation of polypeptides. It leads to the formation of polypeptides or simply proteins. Okay, now so if you take where we can find out the development or the presence of this peptide bond, where we can see means, so first carbon's carboxyl group and with the second carbon or second amino acids amine group, between these two we can observe the peptide bond. So that is somewhat information regarding the linking. Next one, if you continue that thing, so when this kind of a process going to be taken place, so we will observe the elimination of water. So means elimination of water is taking place means definitely we will observe the formation of water. First of all, water formation must be there, then is formed, then only it will be eliminated from the reaction, isn't it? So thereby here that particular process means when two amino acids are linked together, when two amino acids are linked together with peptide bonds, water formation takes place. That's why when water is formed in the reaction, that particular process we are considering as dehydration process. Which one? Dehydration. If water will from the reaction, dehydration. So now only we will know D means removal. So which one is removed? Tanni, water. So that's why dehydration. Next one, very case let us see now. In a polysaccharide, here till now we have seen proteinally what are the kinds of bonds are there, we have seen. In protein, between the amino acid, what is the type of bond is present? Peptide bond. Or what is the name of the bond is present? Peptide bond. Where? In proteins. Now let us see in polysaccharides. What is that? In polysaccharides. In polysaccharides, what are the types of bonds are present, or what is the name of the bond which is present in the polysaccharides? Now let us see. Now polysaccharide. So polysaccharide, the individual monosaccharides are linked by a glycosidic bond. See, they are very, very important. Suppose if you take polysaccharides, polysaccharides means carbohydrates. Likewise, in previous video we learned many monosaccharides, so there are more number of monosaccharides are linked together with a glycosidic bond and results in the formation of polysaccharides. Poly means many saccharides means many carbohydrates or many sugars comes together with the glycosidic bond and results in the formation of polysaccharides. Easy only, no? Means their individual monosaccharide ಏನೋ one monosaccharide and ಪಕ್ಕದಲ್ಲಿ one more monosaccharide and ಹಂಗೆ ಪಕ್ಕದಲ್ಲಿ one more monosaccharide. Likewise, there are more number of monosaccharides, all are comes together and results in the formation of polysaccharides. ఏమో కరెక్ట్ ఓన్లీ నో ఇప్పుడు మోనో 1 అండ్ పక్కతల్లి 1 నెక్స్ట్ 1 ఎల్ఎంఏ కమ్స్ టుగెదర్ అండ్ విత్ గ్లైకోసిడిక్ బాండ్ అండ్ రిజల్ట్స్ ఇన్ ది ఫార్మేషన్ ఆఫ్ polysaccharides. Don't forget why I am explaining this many times means in previous NEET examination they asked this bit. Okay, so that's why. Next one, so then here what is the type of process taking place, sir? Means once you see here again here dehydration process only taking place, means here water is eliminated. Okay, so that's why here once you see this bond also is formed by the dehydration process. So this bond is formed between the two carbon atoms of two adjacent monosaccharides. So between the monosaccharides, so definitely we will observe the formation of glycosidic bond when between the two monosaccharides, when glycosidic bond formation is taking place, at the time water is eliminated. So when water is eliminated, now that particular process we will be considering as dehydration process. Sorry, let us move forward. Now if suppose the next case this third example madri. First of all we have seen proteins, polypeptide chain, no? Next one we have seen polysaccharides, and third one now we are observing nucleic acids. In a nucleic acids or in a nucleic acid, phosphate moiety links phosphate now having reactant nature, or it has the capability or it has the ability to react with pentose sugar. In the pentose sugar structures we see, no? So it can react with that. So that is information only. Let us see here in a nucleotide or in a nucleic acid, a phosphate moiety links with third carbon of pentose sugar or third carbon of one sugar of one nucleotide and to the fifth carbon of pentose sugar of the succeeding nucleotide. Adi you cannot understand. I will explain based on the diagram once you see. Here once you here a diagram you see there. This is pentose sugar only, no? You can recognize this one. Pentose sugar means here we will observe the presence of 5 carbons, isn't it? There 1, 2, 3, 4, 5th carbon, and this 5th carbon of a pentose sugar reacting with the phosphate, and that side you see here one pentose sugar is there, that one third carbon is reacting with the phosphate. So thereby here once you see, if you take one nucleic acid, phosphate moiety links with the third carbon of one pentose sugar, here third carbon only, no? Third carbon of one pentose sugar with the 5 carbon of another pentose sugar. So between these two, here third carbon and here 5th carbon, between the third carbon of one pentose sugar with the 5th carbon of one pentose sugar, when one phosphate moiety is going to be reacted, so there one particular bond formation takes place. Point purida. So now what is a bond name? No, here you see. So the bond between the phosphate and hydrogen group or hydroxyl group of sugar is known as ester bond. So between the phosphate and between the pentose sugar we will observe the formation of ester bond. So we will observe the formation of ester bond. So now there is one such ester bond on either side of it. So means here there is one such kind of ester bond. So here one ester bond and this side also is one ester bond. Together we will be considering as phosphodiester bond. Which one we can call? Phosphodi. Di means 2. So bi means also 2 and di also means 2. So thereby here when we will be observing the presence of 2 ester bonds, so one is here and one more is here, together we will be considering as phosphodiester bond. So phosphodiester bond means here if you take If you take a nucleic acid, no, exclusively we are learning the presence of phosphodiester bond, and if you take a polysaccharides, no, here we are understanding the presence of a glycosidic bond between the two monosaccharides, and if you take polypeptides, no, between the polypeptides or between the proteins, no, we are observing the presence of peptide bond. So here, so this much of information. So suppose in case of polypeptides or in case of proteins, no, between the two amino acids we are observing the presence of peptide bonds, and between the two between the monosaccharides of polysaccharide, no, we are observing the presence of glycosidic bond. And if you take nucleic acids, there between the two nucleotides, between the two nucleotides we are observing, between the nucleotides, no, we are observing the presence of phosphodiester bond. So this is a small information regarding the bonds. And before that one, in a nucleic acid point of view, so what is information we have discussed now? So there are two different types of nucleic acids are present. One is DNA and one more is RNA. So RNA is a first genetic material and which involves in the development of DNA. So now here the DNA is not first formed genetic material. DNA is formed from the RNA, and DNA is nothing but deoxyribonucleic acid, and RNA is nothing but ribonucleic acid. What is the main important differences means, in case of pentose sugar, if you see there, or pentose sugar, if you see there, no, second carbon of pentose sugar consists of a hydroxyl group or simply OH group in RNA, whereas in a DNA point of view, a pentose sugar consists of only hydrogen. There in a second carbon of a pentose sugar, no, we could not find the presence of oxygen. Why? Because that has been removed. So because of that reason only, DNA we are calling as deoxyribonucleic acid. Next one, initial information, if you see there, no, nitrogen bases over there. There are 5 different types of nitrogen bases over there: adenine, guanine, cytosine, thymine, uracil. So thymine is present in DNA and uracil is present in RNA. So this is information, so we can learn in this video. So somewhat information to be continued under nucleic acids. In the next video definitely I will come with that information. Until then, take care and learn the knowledge and be safe at home. Thank you.